Hi everyone, welcome to the Bay Area Science Festival. Specifically, you are joining Can You Believe Your Eyes with the Truhlsen-Marmor Museum of the Eye and Smith-Kettlewell Eye Research Institute. Specifically from that institute, we're going to be joined today by Dr. Preeti Verghese. So we're going to be doing some experiments today. What you're going to need at home if you would like to do these experiments alongside with us, some fingers. You're going to also grab a book. Now would be a good time. Ideally, this book will have some sort of image on the cover or some text or something like that. And then if you want to run to your bathroom or the trash can in your bathroom, grab some, or just a single toilet paper tube. I am curious if it will work with toilet paper still on it. You're going to have to let us know by using the comment section below if that's the type of toilet paper tube you're going to use, but a clean one that is short also works. Or you might be able to get some paper and just roll it up in a tube, but just make sure it's a little bit of a short tube. Okay. So I am pleased to be presenting today with Smith-Kettlewell Eye Research Institute for the Bay Area Science Festival here. Just to let you know a little bit about them, their mission is to create a unique environment for research on human vision. Basically scientific research to increase the understanding of normal vision in support of vision disorders, clinical research to develop new diagnostic procedures and treatments for visual and other sensory disorders. And they also do engineering research to produce better techniques for aid and rehabilitation of the blind and partially sighted. Quickly, I'm going to talk a little bit about the museum while we wait for everyone to join. So the American Academy of Ophthalmology decided to open its doors and show you its renowned collection of artifacts by creating a museum. And this new Museum of the Eye is the world's only free public museum dedicated to the fascinating science of sight. Once you can actually come in to our physical space instead of joining us virtually as you are here today, you'll be able to look through the pupil of an enormous eye. You will be able to explore interactive displays and even try on a virtual reality headset. So through these interactive displays and the high-tech exhibits, you'll be able to see how new technology can impact medicine. You'll be able to discover surprising facts about how your vision works, as well as understanding the field of medicine that is devoted to protecting it. So with that, I think we have a couple extra minutes just to let everybody log in. We are excited to be able to show you a preview of the space. Your eyes are going to be the first to see this, and it's a short 2-minute video of a basically walkthrough of the museum. So I hope you enjoy. was enjoyable to get a sneak peek. Again, you were the first to be able to see that. I just want to remind you that you're always welcome to ask us questions. There's the chat box down below, but then there's also the Q&A. And so we're going to be going through the Q&A a little bit later. And if you type in your questions there, we'll be able to grab those and actually answer them together. So go ahead throughout the program, ask those questions, but know there will be certain times where we're going to actually focus on answering those questions. Okay, so today we are joined by Dr. Preeti Verghese, who runs her own lab at Smith-Kettlewell. So Dr. Verghese, could you tell us a little bit about yourself and what you do in your lab?
Sure, I can try. So I am a senior scientist at Smith-Kettlewell. I've been there for 24 years. I love what I do. I study human vision, both in health and in disease, and try to understand adaptations that individuals with poor vision due to disease can make to take advantage of their residual vision. I mean, it might not be as good as completely healthy vision, but there might be some sparing of vision, and I work particularly in trying to see how to best take advantage of this residual vision.
Oh, well, we're really lucky to have you join us today, especially, I don't know if you know this, but this is the museum's first public program that we're
I didn't know that. That's
cool.
Yeah. When the Bay Area Science Festival first mentioned this, I said, Museum of the Eye?
Mm-hmm.
Oh, I didn't know there was a Museum of the Eye, but
now we're brand new. Yeah, and it's great because we're in the same city too. So eventually, hopefully, we'll be able to be together in person instead of
when all of this is passed. Yes.
Okay, just to give a quick summary about what we're going to be exploring today. So we're going to be looking at how your eyes are just the beginning of your complex visual system. So your brain plays an important role in the visual system, but it can sometimes be tricked into seeing things that aren't necessarily there. So first we're going to explore the anatomy of your eyes, and then talk about binocular vision, depth, and the eye-brain connection. So, I wonder if you could tell us a little bit more about the brain's role in how we see in the eye-brain connection.
Okay, so the eye, you can think of the eye as a window to the world, and the actual interpretation of the image happens in the brain. Now, we wouldn't be able to see if we didn't have eyes, and we wouldn't be able to see if we didn't have the part of the brain that is connected to the eyes. So both of these parts are essential, and the brain has the ability to interpret the image, and I hope we'll have an opportunity to look at examples of how the brain interprets the image. Actually, there's one right here. So we see in this picture kind of an image of somebody looking at a giraffe, and the image of the giraffe appears flipped vertically in the person's eyeball. And you can see that we don't see, if you look at a giraffe in the real world, you don't see it upside down, you see it right side up. And that's what the brain does. The brain actually takes that image and interprets it. And maybe if we have time later, I could talk about how we could mess around with that interpretation. But I think Stephanie has some important other questions to ask.
Yeah, I mean, I am just so curious that the eye actually sees the image upside down, and then the brain does the reflipping of it.
Actually, that part of it is just kind of from the physics. So if you were to make like a pinhole camera and look at, point the camera at something and look at the image, it is inverted. So this is just from the optics. The optics basically turn the image upside down. And so that's the image of the on the retina at the back of the eye, but the brain then interprets that image. And as I said, I'm hoping we'll have examples of how the brain does a lot of interpretation.
Yeah, I wonder if this picture also helps to look at how the visual field is entering into the brain.
Yes, so you can see the eyeballs kind of a third of the way down from the top, and you can see a left eyeball and a right eyeball. There are many labels on this, but I think the purpose of this figure is to say that from the eyeballs, you have these optic nerves leaving the eye and kind of heading down. And you notice that they kind of cross, that's the optic chiasm. Then they stop in a nucleus kind of in the middle center of the brain. And then they go, the first part of the visual cortex that they reach is called primary visual cortex, and it's at the back of your head. And so you see these, I can't point, can you point out the pathway? So leaves the eye, kind of crosses there, stops at that nucleus there, kind of called the lateral geniculate nucleus, but that's not important for us. What I want to point out is from there, it goes to the back of the brain, and this is the part of the cortex, or you know, the largest part of our brain that gets visual input. That is input from this optic nerve that leaves the eye and comes to the brain.
Yeah, I also find that so curious that the eyes are at the front of the head, but the start of that interpretation is all the way at the back, that it has to travel all the way through like that.
Yeah, that's a very interesting question. I should look into that. I don't know how the primate visual system evolved. I should look into that. I don't know. I mean, you know, for example, if you, the olfactory lobe, that is your sense of smell, has a much shorter thing. It's right at the bottom of the brain. And it's a much shorter path from the neurons that kind of sense smell to, so if you look at a rat, it has a huge olfactory lobe and a fairly small part of its brain devoted to vision. And by that you can tell that the sense of smell is a much more important sense for the rat than its vision.
Oh, that's so curious. Yeah. Well, I guess more things that I'm curious about. So most people have two eyes. I can see you have two eyes and I have two eyes. So I'm wondering if you could talk a little bit about how we have two eyes and we have two different views from those two separate eyes.
Yeah, I'm not, I think many people are surprised that each eye has a slightly different view. Yeah. And I think we could maybe show how that works. So I was thinking that if we got participants to pick an object in the room.
I'll do this along with you too.
Okay. Yeah. So we please pick an object in the room. Okay. Close one eye. If you can't kind of keep it closed, you can hold it closed. Okay. Now,
does it matter which eye I close?
It doesn't matter which eye you close. Okay. And then point to that object. So I'm pointing at a pot.
Okay.
And my forefinger. I think my forefinger is aligned with the pot. And now I'm going to open my eye and look at it with the other eye.
Oh, and I'm going to close this one.
Yeah. Is your finger still pointing at the center of the object that you picked?
No, it's so weird. Everything's moving around.
Okay, so I did that badly. So I should have asked you not to. So pick your object again.
Okay.
Point to it.
Yeah.
Keep one eye closed. Now, open. Oh, sorry. This is hard to do when I'm holding my
It's hard to describe, right?
eye. Try to open the other eye and close the eye that you first had open.
It's like keeping one eye open at the same and closing the other and kind of blinking one eye to the other.
And you can alternate it. You can alternate, opening and closing the two eyes. And you can see that your finger that seemed aligned with the object when one eye was open is terribly misaligned when the other eye is open.
It's really like, I feel like I'm jumping back and forth, but staying completely still.
Yeah, exactly. That's exactly it. That's exactly it. So your eyes are laterally displaced. So the view you're getting when you do this with this eye is the same view you'd get if you moved your eye by the same distance, and you see that's a different view.
Oh, you're right. Huh. And just in moving your head.
Yeah.
Yeah.
So,
yeah, it would be this way because I have this eye closed.
Yeah.
So now, like my eyes are all like crisscrossed. Why does that happen?
Well, it's like if imagine if I had a camera here,
yeah,
and I looked through it, and I had another camera here, and I looked through it, I would get a different view. Imagine, so our eyes are only this far apart. Imagine a rodent whose eyes are on the side of the head. They get very different views, completely different views. They get views of that on this side and views of this on the other side. Of course, it increases what they can see because they are prey, and they can keep track of what's around them.
But
so.
if I guess what I'm still confused about is if I have two different eyes with two different views, I only see one image.
That's your question. The reason we have two eyes is to, first of all, increase our field of view, but more importantly for me is that they give us a sense of depth. But for that, the brain, see, we have two eyes that look at the world. The images from the two eyes need to be combined, and they're combined in the brain. And do we have something to kind of, oh yeah, we were going to do that hole in the hand kind of.
Yeah, do you want to go to that?
Yeah, let's do that. Let's see if we could convince people that these two images are combined.
Yeah, definitely. Okay, so now earlier I said go to your bathroom or your trash can in your bathroom and grab one of these toilet paper tubes. Now, for that.
Okay, so we both have our toilet paper tube. Okay, so now I want you to take this tube and put it in front of one eye and look through it. Look at something, for example.
Okay.
Now take the other, take your hand. Take your hand and put it really close to the other eye, like almost touching your nose.
Okay. Like this? Yeah.
Almost touching your nose. What do you see? Do you just see the view through the tube? Do you see your hand? Do you see both?
It kind of looks like there's a hole in my hand.
So for me, I see I see the view through the tube, but around this view is my hand. So it's like I'm looking through a circular opening in my hand.
Yeah, that is crazy. It's like, it literally is like I have a hole in my hand. I feel like it's hard for me to see if like my hand's actually touching my nose, but like if I back my hand up a little bit farther, that's when I start seeing the hole in my hand.
Okay. So let's see if that works for me. Make sure.
It's like it cuts into my hand. Oh, it's so strange. Wait, so what's going on here? Like why?
Okay. So each eye is seeing a different image. And the brain says, well, there are two images coming in, and of course, this is an unnatural situation where you have two completely different views of the world, one through a tube and one with just hand in front of it. But the brain just decides that it needs to put these two images together. And the only way it can do that is to say, well, you see clear something through this tube, and there's where you don't see that is hand. So I'm going to, you know, interpret this as you're seeing
things together.
these things together.
Yeah. Huh. Oh, that's, yeah. Our brains are weird. That's amazing to think through that.
So, so you were asking me, why do we have two eyes? And I said, well, we have two eyes to get two different views of the world. And these signals go up into the brain and the brain combines it. But just because I didn't get any sense of depth to looking at this, it just felt weird.
Yeah.
So I kind of want to convince you that the combination of the two images from the eyes gives you a powerful sense of depth, and that very often this is a cue that we rely on in everyday life when we are doing, like, you know, simple tasks that require coordination between the eye and the hand. And
there's another experiment that talks about
that one.
Yeah, that's really fun.
One too. Sure. So now this is a question of how you get a sense of depth. Okay, so from two eyes. So remember, keep one eye closed. So we're pretending we don't have two eyes. Okay. And hold your hands about a foot in front of you like that. Oh. And point your fingers towards each other. Okay. And then with just one eye open, try to align your fingers so they are pointing but not touching. Do not touch. Okay. So that they're pointing towards each other.
Okay, so I'm keeping space between my fingers, but I'm having them point at each other.
Trying to make sure that they are aligned.
Okay.
You're doing this with one eye open and one eye closed. Okay. Now, open your eyes and look and see how aligned or not aligned your fingers.
They're so not aligned. How do I show this?
Here, they're my fingers.
They're like this.
Sorry, you can't see. Those are my fingers. Yeah, not aligned. Not aligned at all.
What? Oh, hold on. Okay, so if I do that with the other eye, is it just a problem with one eye? Like,
why don't you try?
Okay, I'm going to close my right eye this time and then open it. It's just as off. Well, wait, so can you explain why that misalignment's happening?
Well, so what we're doing is we're for most of us who, you know, who rely on depth, what we're doing is we kind of use depth when both eyes are open. We get to how we see a sense of depth. And that's what we use to align it. But when we don't have a sense of depth, what we're trying to do is say, we're trying to estimate that sense of depth from just the image. And that might not work so well. So you're just trying to estimate, did my fingers maybe look the same size with one eye? If they do, then maybe they're the same distance away, but you're not really getting a sense of depth. And I think the video that we have queued up might actually show that in spades.
Sure thing. Yeah, let's get to that video. We'll click over to it. So this video is specifically from your lab, correct?
Yeah.
Yeah. And so I actually, when I was listening to it, I realized it was your voice giving the instructions.
Yes.
I recognize.
Okay, so this is, before you play it, I just want to, so this is a simple pegboard, a child's pegboard. And we wanted to figure out how easy it would be to kind of place pegs in a pegboard when you only had one eye open. But before that, we wanted to do it with both eyes open. And we did a particularly important manipulation. We put that, you notice that pegboard is kind of a few inches below eye level, but it's not on a table that the participant can look at.
Yeah, it looks like it's aligned with kind of
their it's kind of, you know, at mouth level or something like that. You know, the board is at mouth level. So I believe, so there's no cover over her eyes. So I believe this is the case where she is looking at it with both eyes open. So could you play the video?
Yeah, definitely.
Look, go. Okay, so this was part of a study. You saw she did a great job, effortless. It was part of a study where we were actually tracking the motion of the hand. You saw some sensors on the fingers. We're tracking the motion of the hand, and so we could actually measure how quickly you pick up the peg and place it in the hole. Okay, so do you have the other one where one eye is closed? Okay, so now she's wearing a patch over her left eye, so she's only viewing with her right eye. And I'm going to ask her to do this exact same task.
Okay, so just one eye is covered, but she can still see with the other eye.
She can see with her right eye, yes. Look, go.
Oh, that looks a lot harder, just with one eye closed. You would think you would still be able to see, like you have an eye open, but.
Yeah, so she could see where the hole was, but she couldn't judge its depth. So she misjudged the depth in one direction. And then she said, okay, I'm going to compensate. I'm going to put it the other way around. And she misjudged the depth again too much. And then finally she figured out where to put it. So do you have like a book or something lying around? Whoa. I have a book too.
Oh, okay. So now if you're watching along with us, you can grab your book as well.
Yeah. And then kind of put it at mouth level.
Okay.
In front of you.
Like under my nose, kind of?
Under your nose.
Yeah.
And close one eye.
Okay.
Any eye. And try to point to something on the book with your hand. Like I am trying to point to the letter D, which is a big letter D.
Okay, I'm going to try and point to the letter, up here. Okay. And now what do I do? Do I open both eyes?
You can open both eyes.
Oh, I like totally missed the O.
Look at me. You see where I am? Can you see where the is and the D?
Wow. Okay, I'm gonna do it with both eyes open now. And
okay.
I want to make sure that I can actually do this in general. So I have both eyes open. I'm gonna try the O again. Yeah, and then I totally get it.
I'm going to go for the G.
Did it work out for you?
Oh, wait, I have to show you where the G is.
Oh, yeah, there you go. Yeah.
Again.
Wow, that is so curious. Okay, so I close one eye, I hold the book up, and then I try and select the letter and I point at it, and yeah, I miss it pretty far. So does that mean I have something wrong with my eyes, or is it just this depth of field concept that you're explaining?
You did that with only one eye open?
Yeah.
No. It just means that you're used to using both eyes and that you're relying on both your eyes to give you some sense of depth.
Yeah. Could you talk a little bit about the size comparison experiment?
Oh, this is a very simple but compelling illustration of how the brain interprets images. So I think you should see two kind of, simple flower-like images on the left and the right. Mm-hmm. And the question is, that central circle in the middle, is that the same size or a different size between the two flowers? And
to me, they look completely different. Like
to me, the one on the left is way bigger than the one on the right.
Yeah.
And if I were to, I had a ruler somewhere. If I don't have a ruler, what can I do? I can measure it.
Grab a pencil.
Anything, a pencil, let's see. A pencil. Okay, I'm gonna measure it. Oh, okay.
Turn it up to the screen.
Whoa, they're exactly the same size. Okay. And actually, it's not surprising. I mean, what the visual system's doing is it's making a relative judgment. It's saying,
Mhm.
well, this is surrounded by small things. So in the real world, I don't really care about absolute size. I only care about relative size or relative anything. I want to pick the riper fruit. Um, um, so if the fruit is a strawberry, for example, I might want to pick the reddest berry.
Mhm.
So if I'm looking at a little strawberry bush with mostly ripe berries,
Uh-huh.
um, then a red berry on, oh, okay, sorry, an orange berry, something that's not very ripe on that bush, will look much less ripe than the red berries on the bush, and I will not pick it. But if I'm looking at another bush where the berries haven't ripened, that same orange berry will look the ripest. And
Yeah.
and I might want to pick it, and that's just what the visual system is doing here. So among these smaller circles, it's saying you are big. The one in the middle, that one's the biggest. And among these larger circles, that same size circle looks small. And so the visual system's making these comparisons. You know, it's like taking a contrast. You are bigger than everybody on the left, and you are smaller than everybody on the right. And it's a very useful comparison. It's much more relevant than going around with absolutes. You'll see plenty of instances of how the visual system is kind of making an estimate relative to the immediate surroundings.
That is so curious. And it seems like it's both the eye and the brain making that.
Oh yes, this is the brain that's doing it. The image on the eye is the same.
Yeah.
For the central circle on the left and the right.
Yeah, that's so curious how it plays tricks on your eyes and confuses your brain. And yeah. Here, let's take a look at one other check.
Wow, okay, so this one
Yeah.
is so that was a size comparison.
Mhm.
This is a brightness comparison. So the question to ask is, so you should see kind of a checkerboard surface and a cylinder on it, and the cylinder is casting a shadow. But the question that I would like to ask is, what is the relative brightness of square A versus square B?
To me, they look like square A is gray and square B is white, but in the shadow of the cylinder.
Yep, and that's what everybody thinks.
Yeah.
And so, but is there a way we can do an actual comparison?
Yeah, I've got that lined up.
Oh, okay. So all Stephanie did was to obscure the neighboring checks and to just put two cutouts that covered square A and square B, and you can see that they're identical.
This just blows my mind. I'm going to go back to the first one. So yeah. It looks like gray and white. That's kind of a little bit gray, but then they're the exact same color.
Right.
Like
Can you go back to the original and find Stephanie? So we were talking about A and B, and they're actually the same color, but the brain says, well, this check here is surrounded by dark checks, so it must be lighter. And it says, well, it doesn't have to be the same color as A because it's in shadow, and anything in shadow is going to be darker. So it doesn't care about physical identity here in brightness. It says, I need to interpret the world as B is bright because it is bright relative to its neighbors. That's what I care about. Even though B might be the same brightness as another dark check farther away that is not in shadow. And actually, if you go to this site, Michael Bach He has a whole bunch of really cool illusions. And he tries to explain what causes the illusion. Some explanations are simple and some are more complicated, but this is not too hard.
Yeah, this just, oh, it's just so wild. My I don't want to believe what you're saying, but it, like it does make sense.
Yeah, so the brain just cares about local contrast.
Hmm.
I am bright relative to my neighbors.
We're going to transfer into our question and answer section. While I pull up your questions in the question box, I just want to thank Dr. Preeti for being here today with us, and she's going to help answer these questions. And I also want to thank Smith-Kettlewell also for collaborating with us and the Bay Area Science Festival. We're excited to be launching our public programs. So if you haven't already asked your questions, you can go ahead and do so by using the question and answer box, and I'll read a couple of them, and we will see how this goes. So we have one question coming in that asks, what would be some of the reasons why eyes don't work together? Preeti, do you have an answer for that?
Yeah, there might be many reasons why the eyes don't work together. I mean, they're two eyes, and if during development, you know, there is, one eye has a cataract, or one eye is not pointing in the same direction as the other, or one eye has needs a slightly different correction. It's not focusing as well as the other eye. Then the two eyes are producing very different images, and the brain has to deal with these images. If they're only slightly different, the brain can combine them. But when they're significantly different, the brain has to kind of decide. I'm almost speaking as if there were a homunculus in the brain, but I don't intend to do that. I'm just saying that when it's hard to reconcile the two images coming in from the two eyes, sometimes the input from one of the eyes is suppressed in the brain. So the connections from that eye are not as well represented. And that's, sometimes that is called lazy eye. Sometimes it's, a crossed eye where the eye is not pointing in the two eyes are not pointing in the same direction. And, sometimes you can have congenital cataracts or cataracts that occur, close to the time of birth. So that blurs the vision considerably in one eye versus the other. And
yeah, that actually transitions to another question that I'm seeing from someone. I don't know if maybe you have an answer for this. Someone's asking, how long does it take for our eyes to learn to work together when we're growing up?
So they start work. I mean, there's evidence that they start working together fairly early. People have made it's hard to make functional assessments in very young children, but they've shown that the eyes are already working together in humans at around 3 months. But this connection is plastic, and definitely up to the age of 5 or 6 or even 8, it's malleable, and an insult during that time can particularly affect the course of normal development.
Gotcha. Yeah, there's some really great questions coming in here too. So let's see here. There is one question. Let's see if why is an image inverted top to bottom and not flipped right to left? We were talking about optics of the eye. I wonder if you don't have an answer specifically for that one, if we could revisit like why images are flipped upside down inside the eye, and then how the brain flips it right back side up again.
Okay, so they are flipped. They are flipped, because you're aware of the up-down flip because you normally see a giraffe with its head up. You don't particularly care whether the head is on the right or on the left, but they're flipped both ways. So there is an experiment that, we can't do now, but people have done, where they wear these glasses that flip the image. So when you, so now the image is being doubly flipped. The image is upright in the world, the glasses flip it, and the image in the eye is flipped again. So the image in the eye is upright. But since the brain is used to inverting the image that it gets from the eye, because it's making sense of the world, when you start wearing these glasses, at first it appears as if everything is upside down.
Huh.
But then you get used to it. And this is the plasticity, it says, well, that doesn't quite make sense. So I'm going to, and then when you take the glasses off, everything looks topsy-turvy again, because you just got used to wearing these glasses and reinterpreting the image. So when you take them off, and then you adapt back to, the usual state of things. So I don't know if I answered your question.
No, it was along those lines. Yeah, that definitely answered it. And I think something that I'm definitely learning through speaking with you is just how big of a role the brain actually plays in how we see things and how we interpret things.
Yes, yes. So definitely in the interpretation, vision wouldn't be possible if you didn't have eyes. But yes,
Yeah. I'm wondering, someone was asking about eye disorders. Ooh, there's lots of questions coming in. There's so many. Let me see if I can go back to that one. So
I see a question that says, are there eye disorders where your brain doesn't use both eyes? And I think that was an example that we kind of covered. That's a very easy eye disorder to talk about, because the eyes often look completely healthy, and there is a case of the eyes are sending their input, but the brain can't reconcile the disparate inputs and says, well, I have to do something. I have to kind of downweight the input from one of the eyes. So.
Mm-hmm. I don't know if you see this question in the Q&A. Someone asked, how would it look if you had 3 eyes? I love that question. So, I mean, thinking about hole in the hand, that exercise, and we saw how our 2 eyes are overlapped. I wonder, if there's a fun way to think of that.
It depends on where the 3rd eye was.
Here. Or here. Wait, tell me a little bit more about that. What does placement have to do with it?
So we're getting kind of, so if the 3rd eye was in the middle of the 2 eyes, then, we would, I don't know how we would combine images, but it'd probably give us, if you had 3 eyes, you could afford to lose one for sure, and still get depth. Mm-hmm. You would,
Yeah.
So why do 3D films allow us to visualize a flat picture as having depth? Because 3D films are made with 2 cameras taking slightly different views of the scene, and they do exactly what our eyes are doing for us for free. Otherwise, we're just seeing, the view of a single camera, which is like a single eye.
Mm-hmm.
Oh, I did want to say that, I think we mentioned it when we were chatting, that if you close one eye, but you move your head, you're getting a slightly different view. So motion does help us. Moving through the world helps us to kind of appreciate the depth and relative position of objects as well.
Yeah, that makes sense. Most definitely. It makes me think of like birds and pigeons and chickens that move back and forth, but their eyes are on the side of their heads. So are they judging depth when they do that?
I'm not quite sure that they're doing that. It's more of a postural thing to compensate for the way they walk, but I'm not sure that Yeah, they could be getting different views of the world when they bob. So the question is, if they wanted to get a sense of depth, would they stay still and bob their heads? Um, the body still,
Right.
but they bob their heads. They could definitely get different views. So remember, the eyes are on the side of the head, so they don't overlap and they don't kind of give you two views of the same scene. They're getting two views of kind of very different scenes. So maybe their bobbing the head is uh, a good solution to getting kind of depth.
Yeah, yeah. Um, well, I think one other question that just came in was, are there more activities like this that you can do? And the museum certainly has resources on our website for you to look up more activities. And that website is, I'm going to drop that into the chat box right now. It's basically, if you go to aao.org and find the museum, we have a research and resources page. All right, here you go. I just dropped it into the chat box. So we have, yeah, a lot for you to experiment with at home and learning from home, especially while you're sheltering in place. Are there any other remarks before we kind of wrap it up today that you want to go over, Dr. Preeti?
No, I mean,
Yeah.
yeah, there's a very, so I have to just state a caveat. I am a scientist and not an eye doctor, and it would not be appropriate for me to give any advice on specific eye conditions. I can speak in general about an eye condition. And so if I seem to be interpreting your question a different way, it's because I don't want to specifically address one person's case. I'll try to abstract the question and address it that way. So there's a question that says, if a person had normal visual experience and lost an eye as an adult, can their sense of depth be restored? And the question is a very interesting question because it says, we were just discussing with one eye, you can still get a sense of depth by moving your head around. But the presence of the other eye gives you that sense of depth effortlessly. And there are adaptations you can make to regain that sense of depth, but it's not going to be quite the same as if you had two eyes. I mean, if you had three eyes and you lost one, that would not be a problem, but yeah, they didn't build that redundancy into the system, unfortunately.
Oh, well, okay. I am just so grateful on behalf of the Museum of the Eye and Jenny Benjamin, the director of the museum, and myself, Stephanie. I am the museum specialist here. I just want to thank everybody for being here today, especially Dr. Preeti, for all of your expertise and helping us explore and wonder about all of these concepts. I'm going to just go to some final housekeeping to wrap up the program. Yeah, so that's what we're going to do now.
Thank you, Stephanie. It was a pleasure. It was a pleasure. And I'm so looking forward to visiting the Museum of the Eye.
I am so excited to see you in person as well as everyone else. Wrapping it up today, we are so pleased that you were able to join us for the Bay Area Science Festival and Can You Believe Your Eyes? I am going to just share some more fun facts about the Museum of the Eye, just in case you want to stay in touch and maybe come to more of our virtual public programs. Our next virtual public program will be in November, on November 18th at noon. And this is our launch of our new series called Behind the Scenes. And specifically with this first episode, we're going to be looking at the National Trachoma Service. So basically, if you're interested in medical oddities, you'll want to join our museum director, Jenny Benjamin, for the launch of Behind the Scenes. In this new series, we'll examine curious medical objects in the museum's storage cabinets. The first episode will feature a rare set of glass propaganda teaching slides prepared by the National Trachoma Service. Trachoma is basically a bacterial infection that attacks your eyes and has been documented for thousands and thousands of years. So you can learn about how the trachoma epidemic in the United States, specifically between 1912 and 1924, how it parallels to our current health crisis, the COVID-19 pandemic. Our December virtual public program is happening on December 16th. So you can take a virtual seat at our table as we smell and taste our way through the 16th century eye medicine. Clinical herbalist Ryan Cian of Northwind Apothecary will join us to discuss herbal remedies found in the museum's oldest text, authored by George Bartish. This was from 1583. So together we will sift through these Renaissance era medicinal recipes and explore ingredients used to treat eye issues. We hope that you will join us. You can find those virtual programs and save a seat for yourself by going to our website, aao.org/museum-events. And those tickets are available via Eventbrite. And just remember, it is free, but you do need to reserve the ticket so you can get the link. So please do stay connected. Our website has resources for learning from home, everything from coloring book pages to exploring all of these optical illusions more in depth, and just understanding more about how the eye functions. If you want to join our mailing list, we put out a newsletter every month, every other month or so, and you can do so by going onto our website and clicking the tab Museum Mailing List. And then this is Smith-Kettlewell's website. And of course, last but not least, we are all over social media because we enjoy opening our virtual doors to you. So you can follow us at Museum of the Eye on Instagram, Twitter, Facebook. You can check us out on TripAdvisor and give us a thumbs up on Yelp too. So thanks again for being here today with us, and do reach out with any questions. Again, my name is Stephanie Stewart-Bailey. I am the museum specialist here at the Museum of the Eye, and I definitely welcome any question that you might have. Take care, and thanks again.